Hello and welcome to Prairie's Got the Goods Week. My name is Caitlin Rose Seiler and I am the Stewardship Coordinator with Saskatchewan Prairie Conservation Action Plan, or PCAP. Today, Destiny McGee will be talking about her master's research. How does reconstructed prairie and wetland affect carbon sequestration and weedy invasion in agricultural landscapes? I'd like to start by stating we respectfully acknowledge that we are on the traditional territories of many Indigenous nations and communities, past and present. For millennia, they have worked to protect these landscapes and the life these areas sustain. I would like to thank these original caretakers and acknowledge the ongoing work and presence of Indigenous peoples in Canada today. Before we get going, I'd like to mention that this is our final webinar for Prairie's Got the Goods Week, but never fear, PCAP has another webinar series about prairie conservation called the Native Prairie Speaker Series, and you can register for these webinars on the PCAP website. Just click on upcoming events and then Native Prairie Speaker Series, and we have an excellent lineup for this spring. All past webinar presentations from Prairie's Got the Goods Week and our Native Prairie Speaker Series can be found on the PCAP YouTube channel, and this webinar will be available there in the near future as well. A reminder to our listeners out there, if you have any questions during the presentation, just type it into the questions section of the webinar dashboard at any time. And if you're on a cell phone, you can send your question by chat to the organizer, and questions will be answered at the end of the webinar. I'd like to take a moment to note that financial support for today's webinar and Prairie's Got the Goods Week is provided by our presenting sponsors, Enbridge and Wildlife Habitat Canada. Our supporting sponsors are Eco-Friendly West, Information Services Corporation, Saskatchewan Cattlemen's Association, and in-kind support has been provided by Burr Oak Land Trust. So without further ado, I'm pleased to introduce today's presenter. Destiny McGee received her BA in Natural Resources and then her Master's of Science in Ecology at the University of Northern Iowa, UNI. While pursuing her BA, she worked with the United States Geological Society, USGS, to survey fields enrolled in the Conservation Reserve Program, CRP, which led to her graduate research at the University of Northern Iowa. Destiny has always had a special interest in the ecosystem services of native vegetation in highly altered landscapes, and currently works with the Burr Oak Land Trust in Iowa City, Iowa, administrating land management activities on native prairie, wetland, and oak hickory woodland. So with that, I'd like to pass it over to Destiny.
Great, thank you, Caitlin, and hello, everyone. Here we go. Okay, so as Caitlin said, what we're talking about today is based on my master's research. And this research was part of a larger research project run by the United States Geological Society, or the USGS. And the objective of this larger research project was to find a way to quickly and accurately survey the Conservation Reserve Program and determine how successful it was in reaching the goals that were set out for it. And I was part of this research, and then I took it a step further to look at how the fields enrolled in this program were affected by weedy invasion and if they were serving as a carbon sequestration service. See, that's not going on. There we go. So I want to give you a little bit of outline of how I'll be presenting this research to you. First, I'm going to give you a little background on CRP and what it is. And then we'll move into my specific research topics, which were weedy invasion and carbon sequestration. So I'll start each of those by giving a little intro. I'll go into the methods that we used and then the results that we got from that research. So as this research was centered around reconstructed grassland, prairie, and wetlands that were part of the Conservation Reserve Program, or CRP, one of the largest private land conservation programs in the United States, I want to give you a little background on what that entails. The CRP program has been around for almost 40 years, and it was started back in 1985. And it's overseen by the U.S. Department of Agriculture, or the USDA. And this program allows landowners to take environmentally sensitive agricultural or pasture land out of production for a contract period of 10 to 15 years and plant them with perennial vegetation in exchange for a yearly payment. And as you can see on the chart on the right here, the peak enrollment for this program was around 2007, when there were almost 37 million acres enrolled. But currently it stands around 22 million acres. So this is a loss of about 15 million acres of grass and wetland from the landscape. And to give you an idea, that's equivalent to about 10% of the Saskatchewan land area. So it's a lot of acreage going out. So although this program puts habitat back on the landscape, there are large fluctuations in the acreage, with many of these expired CRP fields likely reverting back into agricultural production. So this figure on the left shows the CRP enrollment in the United States in 2020, and the amount of acres enrolled in the program are pictured by color. Where I did most of my research is here in the Midwest US, also known as the Corn Belt. And as you can see, it's mostly red, which represents 10 to 50,000 acres that are enrolled in CRP. In particular, you'll see Iowa, which is right in the center of that circle there, is almost completely red. And that's because about 86% of the land cover here is actually in row crop production. And the highest amount of acres enrolled in the program is represented by the yellow, and that's greater than 50,000 acres enrolled. So if you look at this yellow band here, this is the exact area in the US that was hardest hit by the Dust Bowl era back in the 1930s, when those drought conditions and exposed soil created those large dust storms. And this isn't entirely surprising that there's so much CRP here, because that's actually the issue that the CRP program was started to help alleviate. So the CRP program has evolved over its history, but as I said, at its conception, the primary goal of the program was to curb soil erosion due to agricultural practices. CRP helped curb this issue by establishing vegetation cover on and around at-risk land, for example, creating contour strips. And because erosion prevention was the primary goal, the USDA did not have any requirements for the kind of perennial grasses that were planted. So the first practice was mainly brome monocultures, as you see in the bottom picture there, which is a non-native invasive grass. And this was partially because restoring grassland was not a goal of the program initially, and also because there was almost no commercial native seed market in 1985, so there really was no other viable options at that time. So the first step in using this program as potential for grassland habitat, which as a conservation practice, abbreviated to CP, was CP2, in which they required that native species actually be planted in the field. And since then, the program has evolved to include other goals, including mitigating water quality issues with wetland restoration, as you see in the top picture there, that's CP23, and also grassland reconstruction and in creating wildlife habitat. Most recently, CP42, which is the practice for pollinator habitat, is the most wildlife-oriented practice to date, as its goal has nothing to do with erosion control and everything to do with the bee and monarch butterfly declines. So this program actually requires a certain amount of pollinator-friendly forbs to be planted. And in case you haven't heard this term before, a forb is just an embraceous, meaning not woody at all, flowering plant that's not a part of the family of grasses. And all of these conservation practices that I have listed here are ones that were included in the research that I did. And as you can see, the goals of the CRP program have changed, and they need to continue to evolve in order to address modern day issues. My research was focused on 2 ecosystem services that are not direct goals of the program, but that can be indirectly addressed, which is biodiversity and carbon sequestration. So biodiversity is the first ecosystem service from CRP fields that I'm going to address. In the US, the Midwest ecosystem is dominated by these crop monocultures, as you can see in the left picture there of a cornfield. And when grassland and wetlands are added into this landscape, the vegetation biodiversity is increased, as you can see on the right there, as a picture of a prairie strip in a soybean field. And there's just a lot more species richness there and a lot better habitat. So biodiversity is a central theme of community ecology, and research from this field has shown that high diversity increases the competitive environment of communities and thus makes it more difficult for invaders to get established and succeed. This is because biodiversity protects against invasion by increasing the competition for space, available light, nutrients, and other resources. An important thing to think about when we're talking about invasive species is what is an invader and what makes it an invasive species. So the first requirement for an invasive species to be considered invasive is that it is an alien species, meaning that it's a species that's living outside of its natural geographic range. This doesn't necessarily have to mean that it's from another country. And these species escape the natural barriers that keep them in their natural geographic ranges, usually because of human activities such as travel, where seeds can stick on the vehicles and tires and make it out of those areas where they were originally stuck in. And what makes one of these alien species an invasive species is when it outcompetes or overruns native species, causing serious ecological, economic, or human harm. In terms of ecological consequences, invasive species can create devastating consequences as they can alter entire ecosystems. There are several ways that invasive species can have these negative effects on an ecosystem, including outcompeting those native species for resources, as well as hybridizing with some native species, therefore altering the genetic makeup and reducing the native gene pool of native species. Recent studies have shown that this problem is likely to get worse as global temperatures rise and precipitation patterns change as a result of climate change. In terms of economic damage, in the United States alone, invasive species cause approximately $120 billion in damages and control costs a year, and around $26 billion of these costs are incurred by the agricultural industry. Invasive species are also the second leading cause of biodiversity loss behind habitat loss. In fact, nearly half of the species that are listed as threatened or endangered under the U.S. Endangered Species Act are considered to be threatened or endangered primarily because of invasive species. So let's talk a little bit about invasive species in agricultural environments specifically. Over 500 introduced species in agricultural landscapes have become weedy pests, and of those, approximately 73% are not native species. Intensive agricultural systems, such as those in the Midwest region of the US, can intensify the propagation of weeds. In fact, as the management of an agricultural field is intensified, the greater the potential is for habitats adjacent to those fields to become a source of weeds to those ag fields. This indicates that these reconstructed grassland and wetlands in the form of CRP fields could be at an increased risk of being invaded by a weedy species. A couple of reasons for this is that frequent disturbances and habitat fragmentation create space for invaders to colonize and have been found to decrease the ability of native plants to compete. Another reason for this is that agricultural disturbance regimes are often associated with an increase in nutrient availability, therefore providing less competition for certain nutrients, especially nitrogen. Also, seed and cattle transport can become modes of transportation for the seeds of these invasive species, and it's in these habitats with these characteristics of disturbance and increased resources that most successful plant invasions occur. So as I mentioned before, one of the ecosystem services that CRP fields can bring to a heavily agricultural landscape is biodiversity. While these fields are seeded and planted with native and beneficial grasses and forbs and creating valuable habitat and being beneficial to native species, they can also create a lot of potential habitat for weedy species, since the intensely managed agricultural landscape has many factors that can benefit their establishment. So therefore, as part of this research, I wanted to assess not only the benefits of CRP fields, but potential negative environmental effects, such as promoting the invasion and spread of invasive species, and how native biodiversity might help mitigate that potential. There are 2 weedy species of particular concern in Midwestern CRP fields that I decided to use as the subject of this. They are wild parsnip and Reed Canary Grass. So I want to talk a little bit about why these 2 species. I'm going to start off by talking about wild parsnip. Wild parsnip is a highly invasive forb that's native to Europe and Asia, and it has been spreading across North America. It's commonly found in road ditches and other disturbed habitats, and it has even been found to invade native remnant prairies. This species also causes human harm, causing a condition known as Phytophoto Dermatitis, as you can see in these pictures here. They cause these blisters on people's skins, and this happens when the plant is broken and the sap reacts with sunlight, causing the skin to burn and creating these blisters. Other concerns with this plant are that it reduces the quality of agricultural forage crops, as they can harm cattle when they're ingested, and they can also decrease the habitat quality for honeybees, as honeybees do not use or pollinate parsnip, and it can displace important pollinator plants. There are several factors that make parsnip a good invader. It has a long, deep taproot that grows roots down to 1.5 meters deep, and this helps parsnip with nutrient and water uptake. Parsnip also produces a large amount of seeds that can stay viable in seed banks in the soil for years, contributing to its ability to persist and reproduce, making it a good colonizer species, which is one of the first species to inhabit a disturbed area. While surveying CRP fields around the Midwest, I commonly saw this species invading CRP fields. The other species I used in my research was Reed Canary Grass, which I'm now going to refer to by its genus name, Phalaris. Phalaris is actually native to North America, but it's also native to Europe and Asia. And somewhere along the line, Phalaris in North America hybridized with other species native to Europe and Asia and became a noxious invasive weed that's now present in 43 states in the US. Phalaris is particularly aggressive in wetlands as it grows in thick, dense mats and outcompetes and overtakes other native wetland species and creates these large monocultures. As such, it's considered one of the top invasive weeds invading wetlands in the Midwestern US. Some of the biological advantages that Phalaris has that makes it so invasive is that it develops above ground biomass rapidly, and so it quickly shades out any competitors. It also has 2 modes of reproduction. It reproduces along rhizomes, which you can see a picture of here. So these rhizomes are just underground stems that grow horizontally under the soil and let up a lot of new plant shoots along that stem. And these rhizomes can also store carbohydrates and allow for that early rapid growth in the early spring, outshading native species. Phalaris also reproduces through seeds, and it has a very high annual seed yield, and these seeds can also stay dormant and viable in seed banks, allowing them to take advantage of those newly disturbed environments. Along with wetlands and riparian habitats, Phalaris is also common in roadside ditches, and its small seeds are easily transported. In fact, studies have shown that in wet prairie grasslands, fertilization and disturbance, which are both common in agricultural landscapes, increase both the biomass and frequency of Phalaris presence. So like parsnip, this is a species that I saw commonly in CRP fields. So CRP fields are used to bring the biodiversity and all the ecosystem services that come with them to agricultural landscapes that would otherwise consist mostly of large monocultures of crops. While these fields can be beneficial to native species and increase the biodiversity, they also create a lot of potential habitat for weedy species such as Phalaris and parsnip to establish in a landscape that has many factors that benefit their establishment. Therefore, they have the potential to invade and degrade these reconstructed prairies and grasslands in the form of CRP. And the CRP should be assessed not only for the benefits that they bring, but also for potential issues such as the invasion by these weedy species. So these here are the specific questions I aim to answer with my research. First, I wanted to know, do Phalaris and parsnip invade specific CRP practices more frequently? And does the invasion success change with the age of the CRP establishment? Second, I wanted to know, does disturbance or vegetation characteristics such as the grass and forb richness, which is just the numbers of species of grass and forbs, or the percent cover, or how much bare ground there is in a field, predict the invasion of Phalaris and parsnip in CRP fields? So in order to answer these questions, I used data that was collected as part of the larger research project I mentioned earlier. In this research, edge of field surveys were used to visually assess 1,793 CRP fields in the 14 states that are listed here. And if you look at the map over here to the left, all those blue circles there are a CRP field that was assessed as part of this research. The goal was to survey at least 20 fields from each conservation practice, and I personally surveyed the fields in Iowa, Minnesota, and Missouri, so right around this area here. But all the surveyors use identical protocols and data collection forms. At each of these edge of the field surveys, we looked at the forb and grass species visible from the edge of the field, as well as each species percent cover and the overall percent cover of forbs and grasses for the entire field. So these surveys were done from the edge of the field because CRP fields are still private property, and in order to get onto the land, you need landowner permission, and this would take a lot of time and effort to acquire. Another reason, as I mentioned before, is that the USGS is looking for a way to do a rapid assessment of several CRP fields, and as you saw on the first slide, there are thousands of acres of CRP land in the United States. In order to validate the effectiveness of these edge of the field surveys, we actually went into 68 CRP fields and did an in-field survey from 3 different states. And at each of these locations, we did that edge of the field survey followed by an in-field survey. The in-field surveys consisted of a detailed sampling of the vegetation, including the same parameters as the edge of the field surveys. We sampled vegetation in the field by laying down 4 trend sects, which are these lines you see on these figures to the right, and took vegetation samples from 4 quadrats, which are these little yellow circles on those lines, and for a total of 4 transects in each field. And we chose one of these patterns that best suited the overall vegetation of that field. And here's a picture of a quadrat. So we would take all the vegetation that was in that little square meter and record that. So moving on to some of the results. This is the result from the in the field validation surveys that were done in order to determine if the edge of the field surveys were doing a good job of determining the vegetation in the fields. So on the left here, we have the forb cover estimate, and on the right, the grass cover. And on the x-axis there, you'll see that's the roadside, the edge of the field estimate. And on the y-axis, we have the in-field estimate. So this showed that the estimation of vegetation in the field done on those edge of the field surveys were strongly correlated to what was seen when we were looking in the field, and therefore are a pretty good way to rapidly assess a field and validate this weedy invasion data. So the presence and absence of a plant species can be caused by local factors within its potential distribution range, such as climate and geological factors, which was beyond the scope of this study. So even though these species can be found in areas outside of the Midwest, states outside of this area were left out as they're geologically disjunct from the Midwest. In order to outline the potential distribution range for Phalaris and parsnip, I used the minimum convex polygon method in ArcGIS with the sites where Phalaris and parsnip were present. The CRP sites outside each species distribution range were not included in the following analysis for each species. So the map on the right here shows the fields that were used to determine that minimum convex polygon. And all of these dots on the map are CRP fields that were surveyed, and the red dots that you see are representative of the fields where Phalaris was present. So this green figure here shows what that minimum convex polygon came out. And so that is the range that we used in order to determine this, and those would be all of the fields that are included in the following results. Okay, so I'll move on to talk about the bare soil presence in the CRP and how it affected the presence of Phalaris. If you look at this graph here on the x-axis, we have that bare ground cover percent that was in the field. And on the y-axis, you're going to see the probability that Phalaris was present in that field compared to that bare ground cover. The results here showed that the amount of bare soil present in the CRP had a significant negative effect on Phalaris presence, meaning that fields with more bare soil areas were less likely to have Phalaris present. This was a surprising result for us, as we expected fields with more bare soil areas to be more likely to have Phalaris present, as it represents more space for it to invade. However, when we looked at years in CRP, that had no significant effect on the presence of Phalaris, which could indicate that these fields are invaded early on in establishment and that Phalaris quickly fills in these bare soil areas, creating this large monoculture. We'll move on to the forb and grass cover. So here is two similar figures showing the relationship between forb and grass cover with the presence of Phalaris. And you'll notice that the one with the grass cover excluded Phalaris, so Phalaris was not included as a grass in that cover analysis. These results again here showed that higher percent grass and forb cover both had a significant impact on the presence of Phalaris. And CRP fields with greater forb or grass cover had a significantly less chance to have Phalaris present. So that greater biodiversity was helping keep that Phalaris from being present in these fields. Okay, moving on to the grass and forb species richness. So this is how many grass and forb species were present in the field. So CRP fields with higher grass species richness were significantly less likely to have Phalaris present. However, greater forb species richness had no significant effect on the presence of Phalaris in these CRP fields. This result suggested that increased biodiversity and native plant cover reduce the probability of Phalaris invading CRP fields, and that grasses have a higher impact on this. But as you've seen from the last few graphs, there's still a lot of variability within these results. So although higher biodiversity is a factor in decreasing the likelihood of Phalaris invading a field, it's not the only factor that's going to affect this. All right, we'll move on to it, looking at it by the conservation practice type. So on the x-axis here, you're going to see these different conservation practice types. And on the y-axis is the percentage of Reed Canary Grass that was either absent or present, with absent being shown by the red color and blue being Phalaris is present. So Phalaris was present in every conservation practice type that was surveyed, but it wasn't significantly correlated with any conservation practice type other than CP23, which is that wetland practice, where it was significantly more likely to be present than the other conservation practice types, having been present in approximately 50% of the CP23 fields. This was not a surprising result, as Phalaris are prominent invaders of wetlands and those riparian habitats, but it does indicate that these wetland reconstructions should be monitored closely for potential Phalaris invasion to keep these fields from becoming a single species monoculture and keeping a healthy and thriving wetland. So moving on to parsnip here. In the Midwest, parsnip was present in Iowa, Minnesota, and Missouri, but that delineated range with the minimum convex polygon mainly consisted of Iowa and a portion of northern Missouri. Again, this doesn't mean that parsnip doesn't exist outside of this range. There were 329 fields surveyed in this range, and parsnip was present in 64 of these fields. While parsnip was found in at least one field of each conservation practice type in the CRP program, its presence was not significantly affected by any conservation practice type like Phalaris, meaning that it was just as likely to invade any kind of practice under the CRP program. So now let's look at how bare ground and age of a CRP field affected the presence of parsnip. This figure shows the relationship between the percent bare ground area, again on the x-axis, and the probability that parsnip will be present in a field on the y-axis there. So percent bare soil found in a CRP field showed a significant effect on the presence of parsnip, with CRP fields that had higher bare soil areas being less likely to have parsnip present. Again, this was a surprising result, as we expected to see areas with greater bare soil percent be more prone to invasion. However, the amount of years in the field had been enrolled in the CRP program was again not significant on the presence of parsnip, once again suggesting that parsnip likely invades CRP fields early in their establishment, and that could explain this bare soil result that we're seeing here. So here we'll look at the forb cover and richness effect on parsnip. Again, we have the forb cover on the x-axis here, and we excluded parsnip as a forb in this analysis, and the probability of parsnip being present on the y-axis there. So increased overall percentage of forb cover significantly reduced the chance of parsnip being present in a field. However, the forb richness was not significant, suggesting that the amount of forb cover is important in the resistance of a community to invasion by parsnip, but not the number of forb species. So let's look at the grass species and cover effect on parsnip. In this graph here, you see the grass percent cover on the x-axis and that probability of parsnip there on the y-axis. Parsnip present was correlated with grass species richness in a marginally significant way, so it was just on the cusp of being significant, with parsnip presence less likely in fields with higher grass species richness, but it was not significantly correlated with the percent grass cover. These were once again interesting results, with grass species richness having a significant effect and forb species richness not, especially because increased grass cover did not have a significant effect, but the greater forb cover did. These results again suggest that increased biodiversity and native plant cover reduce the chances of parsnip invading these CRP fields, but it does suggest that the kind of functional groups of grass or forb associated with this increased biodiversity and cover might be important. So I just want to go over the important things I want you to pull from the previous slides. First, we saw that Phalaris invades CP23, the wetland practice, more frequently, indicating that wetland and riparian reconstruction should be closely monitored for invasion by Phalaris, but there were no other correlations between Phalaris or parsnip and conservation practice type. We also saw that an increase of grass cover and species decreased the chances of Phalaris presence in a CRP field, and an increase in forb cover decreased the parsnip presence. This indicates that having that healthy, biodiverse, reconstructed prairie or wetland is a significant factor in keeping invasive and weedy species out of them, and then not becoming a source of weeds to nearby agricultural fields. The presence of both of these weedy species were not correlated with field age, but fields with higher percent bare ground were less likely to have both Phalaris or parsnip present. And this could indicate that those disturbances on the site prior to and during reconstruction create space and opportunity for these two species to colonize the site early on in establishment. This is further supported by the fact that invasive species are expected to begin growth earlier than native species, and that climate change may be increasing this phenomenon. This brings me to that second potential ecosystem service of these reconstructed grasslands and prairies, carbon sequestration. So I'd like to start off by talking about why carbon sequestration is important. Carbon dioxide is at the center of attention when it comes to mitigating greenhouse gases from the atmosphere in order to limit global climate change. One main reason for this is that CO2 comprises around 60% of the total greenhouse gas emissions due to human activities, and it has a long atmospheric residence time, longer than 100 years. An important contributor of CO2 into the atmosphere is land use change. It's estimated that land use change has contributed to over 1/3 of the CO2 emissions to the atmosphere since the mid-16th century. And when I say land use change, I'm referring to the conversion of natural ecosystems, such as prairie or forest, into agricultural or urban development. So why does land cover make such a difference? It is estimated that soils have lost 30 to 50% of the carbon that they once held before this land use change, which makes a significant impact on atmospheric CO2, as soils hold over 75% of carbon held in terrestrial systems. An important example of land use change includes the clearing of natural areas for agricultural uses, such as in Iowa, where over 99.9% of our natural tallgrass prairie land cover has been converted, 86% of which is now devoted to row crop production. So how would CRP fields help sequester atmospheric carbon back into the soil? A major input of carbon into soil is from plant photosynthesis. Photosynthesis fixes that atmospheric CO2 into plant biomass, and 30 to 50% of this carbon is used to build root biomass. The majority of soil organic carbon is based on the biomass of these plant roots. This is because carbon that the plant doesn't use for growth gets exuded through the plant roots. There's also a small amount of soil organic carbon that comes from dead plant litter. However, as there's more oxygen above ground, a lot of this litter is broken down above ground by microbes, which need that oxygen to decompose organic waste efficiently. While some of the carbon deposited into the soil is respired back into the atmosphere from microbe respiration, a result of microbes breaking down organic material, the rest is stored in the soil as organic matter. The soil is very good at storing this organic carbon because it restricts oxygen availability. And as a result, carbon that has been deposited by plant roots and dead litter tends to stay in the soil. And if those carbon inputs from photosynthesis exceed the carbon lost, then soil organic carbon is going to increase over time. The flux of carbon going into and out of the soil is very large relative to the amount of CO2 in the atmosphere. So even a small change in this amount of carbon going in or out can show a significant change in that atmospheric CO2. Recent evidence has showed that a large amount of soil carbon has been lost due to accelerated soil respiration, leaching, and erosion, all of which are increased in agricultural landscapes with practices such as tilling and plowing that aerate that soil and eliminate those low oxygen conditions that slow down the decomposition of soil organic carbon. So as we just talked about, the majority of the soil organic carbon is primarily based on the root biomass. The figure on the left here is showing the roots of different native prairie plants. As you can see, these prairie plants have extensive root systems, both in how they spread out and how deep they grow. They can grow down to depths of 5 to 15 feet, so pretty deep. And on the right here is a picture of a soybean root and of a corn root, which are both common crops here in the Midwest US, which you can see are much more shallow and don't spread out near as much as prairie plants. Because of this, we would expect those native prairie plants like those planted in CRP fields to sequester more carbon than agricultural crops. And this leads me to the specific questions that I was looking to answer with this research. So first I wanted to know, do CRP fields have higher total soil carbon when they're compared to adjacent cornfields? Secondly, I wanted to know, does total soil carbon increase in CRP fields over time? And lastly, I wanted to know, does total soil carbon differ between different CRP practices? So let's talk a little bit about the methods that we use to answer these questions. We took soil samples from 34 different CRP fields, and then also from 5 cornfields that were adjacent to prairie CRP fields in Iowa. The reason that it was important to test a CRP field with an adjacent field is that so that you could see the effect of the difference in those land uses without having to worry about any other variables that could be contributing to the carbon sequestration, such as temperature and precipitation. The CRP fields ranged in age from 3 to 15 years old, but how long the cornfields were in production is unknown. The soil samples were taken in the same way that the infill validation was done, where samples were taken from 2 quadrats on each of those 4 transects. So we had a total of 8 samples per field, but then we took 2 different soil samples in each of these quadrats. One with an oak core, which you can see is the top middle picture there. This allowed us to take soil samples from different depths from 0 to 30 centimeters, and these soils were used to determine the carbon concentration that was in the soil. In the bottom middle picture there, you'll see a bulk density core, which we used in order to test the bulk density of the soil, which is an important factor when you're going to determine the total soil carbon content. So I'd like to talk a little bit about why bulk density is a necessary measurement. While root biomass is an important regulator of carbon sequestration in soils, the properties of the soil itself also have important implication for the ability of the soil to sequester carbon. One of these properties is bulk density. Bulk density indicates the level of compaction that a soil has experienced, as seen by how many air pores are between the individual soil particles, or how fluffy the soil is. So here we are looking for the mass of a soil in a given volume. I have a few examples over here on the right. See the one that says low bulk density, you would see that those soil particles are further apart and there's more space between them. And to the right of that is indicating those soil particles being more packed together with less space between them. And this can influence how well root systems are able to infiltrate and grow in the soil. And it can also influence the availability of resources like water and nutrients. So consideration of this bulk density is important when we're measuring carbon sequestration in the soil, as it has implications for how much carbon is being sequestered per unit volume of soil. Also, because soil composition and carbon concentration can greatly vary, I compared the soil carbon concentration measured in each sample to the mean soil concentration expected for each soil type in the CRP and cornfields. This was done by subtracting from the average measured percent carbon for a given soil type as reported through the USDA soil survey. In addition, the effect of the grassland's age was tested in order to determine if soil carbon concentration increased or decreased over time. So let's move into the results here. So this shows the baseline carbon results, which is when we were talking about when we're comparing it to what we would expect for that soil type and what we actually saw. So in this figure here on the x-axis, you're going to see that conservation practice type, and on the y, it's that soil organic carbon difference. So this red line here is going to be what is expected for that certain soil type. And if you see a dot above that, that's indicating that that soil has sequestered more carbon than is expected for that soil type. And if it's below, it's going to have less carbon than is expected for that soil type. So the analysis of this showed that the average CRP field soil carbon content was higher than expected for each soil type. This was except for CP23 fields, which again are those wetland programs, where they are 1.8% lower than expected. In the CP23 programs, it's required that they have at least 50% hydric soil in the field, and these wet hydric soils are anoxic and therefore capable of storing soil carbon for longer periods of time. And it's likely that these soils would lose a significant amount of carbon after these mechanical disturbances from agriculture, and it would take longer to return to expected soil carbon levels for that soil type. Okay, moving on here. So now we're took a couple different measurements to find the total soil carbon in these reconstructed prairies. We will look at the LOI %, which stands for loss on ignition, which is a method of determining soil carbon by burning off the soil carbon and tell us how much carbon was in that soil sample. But we will also have to look at bulk density to see how much soil was in the sample. And after we look at these two results, we can find the most important indicator, which is how much total soil carbon there is in a soil sample. So I want to bring your attention over to the right of this figure first. Overall, the habitat here is corn and prairie, with the corn being that darker gray and the prairie being that lighter gray. When we look at the soil carbon concentration overall, the reconstructed prairies had a significantly higher carbon concentration than the cornfields did, with prairies averaging around 7.6% carbon concentration and the cornfields averaging about 5.5%, which is a difference of around 2%. Now, if you look over to the left side of that here, these are showing those prairies that were directly adjacent to a cornfield. And on the x-axis here, we're just seeing the site name as an indicator of which site it was. And while you can see that 2 of the paired corn and prairie fields show corn with a higher carbon concentration here under Brundle 2 and Miller, they were not significantly different from each other, and that was likely due to factors that were beyond the scope of this study, such as how the field was used before being converted to prairie. But the other 3 that you see here, the prairies had a significantly higher soil carbon concentration than the cornfields. So we'll move on to that bulk density. I'm going to bring your attention again over here to the right side of this figure, with the corn being that dark gray color and the prairie being that light color. And on the y-axis here, we're seeing the bulk density, which is the grams of dry soil in a cubic centimeter. And when we looked at the average bulk density between all of these reconstructed prairie and cornfields averaged, the reconstructed prairies had significantly lower bulk density at around 1.5 grams of soil per cubic centimeter, and those cornfields had significantly higher bulk density at around 1.3 grams per cubic centimeter, a difference of around 17%. And effectively, this means that cornfield soils were more compact with less space between soil particles, and prairie soils were more porous and less compact. Now, if we look over to the left, again, we see some variation. If you'll notice, it's the same 2 sites, Miller and Brundle 2, that had the lower carbon concentration that also had that higher bulk density. So there's likely something going on with those 2 sites, like how old those prairie reconstructions were. But in the other 3 paired fields, we do see that significant drop in bulk density between the prairie and the cornfields. So moving on to the important results that really tell us what's going on in the soil. This is the important part because we're taking that carbon concentration and that bulk density together to see how much carbon there is in a given volume of soil. So again, if we looked over to the right first, we're going to see the averages between those cornfields and those prairie fields and our total carbon content here on the y-axis. So if we remember from the first slide, cornfields had a lower carbon concentration, but with their higher bulk densities, the total carbon content of the soil was only slightly, but still significantly lower in corn compared to reconstructed prairie, with prairies having about 1.7% more total carbon than those cornfields. Now, if we look over to the left, we see those individually paired fields again. And here we'll see that those 3 reconstructed prairies still showed that increase in total carbon in the soil, and those same 2 that were different in the other 2 are slightly lower. But as we talked about before, this could be for reasons that are beyond the scope of the study, but it could also be because of another factor that we did look at, which is the age of the reconstructed prairie, which is the next result that we'll look at. So here we're going to see the site age on the x-axis here, and I want to bring your attention first to the top graph here, which is showing us that carbon concentration that was in the soil. And as you can see, there's a trend there that shows that carbon concentration increases as the CRP increases in age, with the oldest prairies age 15 years showing the highest carbon percent, and prairies age 6, which were our youngest, having the lowest, showing a carbon percent increase over the life timeline of around 3.4% to around 10%, which is an increase of about 6.7% over that 9-year time difference between the youngest and the oldest. Now, if we look at this one directly below that, this is going to show us the bulk density in terms of years. So bulk density averages steadily decreased with increased CRP age, decreasing from about 1.6 grams of soil per cubic centimeter in those youngest prairies to 1.1 grams of soil per cubic centimeter in the oldest prairies, showing a decrease of about 0.5 grams of soil per cubic centimeter. So they're getting fluffier and fluffier as they're getting older. Now, if we look at the bottom, that's the important thing that we want to look at when it comes to carbon sequestration. It shows that there is a steady increase in that total carbon content as the CRP field increases in age, with the youngest having the lowest total carbon content, and as they age up to that 15 years, they're going to have a lot more total carbon. This shows a carbon sequestration rate of about 6.4 milligrams of carbon per cubic centimeter in the top 15 centimeters of the soil in these CRP fields. So as we said before, even a small change in this is going to make a significant difference in that atmospheric CO2. But again, these results likely depend on the time since prairie establishment, the soil type, and the history and management of both the fields of corn and prairie, explaining how we saw those variants in the comparison between the paired corn prairie fields. Furthermore, evidence of past agricultural management, including higher bulk density and lower percent soil carbon, can be seen in restored prairie soils more than 50 years after the disturbance. So it really takes a long time for these prairies to go back to what they originally would have had in the soil. And because past management histories of our sites were unknown, this may have contributed to some of that observed variability as well. So here's the important takeaways I want you to take from the carbon sequestration part of this. Total soil carbon was significantly higher in these CRP fields when compared to adjacent corn fields, indicating that these reconstructed prairie, wetlands, and grasslands in the form of CRP have an increased carbon sequestration rate over those agricultural fields. CRP fields also showed that increased total soil carbon when compared to expected soil carbon of those individual soil types, indicating that only do CRP fields provide more carbon sequestration than agricultural fields, but they're also providing an increased carbon sequestration than is expected for a given soil type. I would also like to point out that CRP fields not only increase that total soil carbon, but they're also decreasing that soil bulk density, and thereby they're really improving that health of that soil. And these are the two takeaways I would like you to take with you. First, that the increased biodiversity in these prairies, grasslands, and wetlands really helps protect against unwanted weedy invaders from the landscape in ag fields and in our natural native habitats. And the second thing I'd like you to take along with you is that these restored and reconstructed grasslands and wetlands can be a really important carbon sink when it comes to reaching goals to reduce that atmospheric carbon that causes global climate change. Lastly, I'd like to give a little acknowledgement because I had a lot of help with this research and couldn't have done it with all of the assistants and collaborators that I had with me. So just wanted to take a minute and acknowledge them. And then I'll take any questions that anyone may have.
Awesome. First of all, thank you so much for the great presentation. It's really nice that you're able to explain your master's research in ways that people can understand. So thank you for that. I know we're a little tight on time, so I'm going to kind of, oh, that's okay. That's okay. It was a great presentation. I'll just sort of give you the questions rapid fire and we'll see how many we can get through. So the first one from Ross, what depth was the carbon sampled?
So we went down to depths of 30 centimeters. Originally, we had thought to look at it in different samples from 0 to 10 or 10 to 20, but somewhere along the lab, the soil samples got mixed up. So we weren't actually able to look at the difference between those 0 to 10, 10 to 20 horizons.
Okay, and would not the depth of the topsoil have significant impact on carbon retention?
What was that?
Would the depth of the topsoil have significant impact on carbon retention?
Oh, sure. So where the significant carbon is going to be being sequestered is where those roots are going to be from the plants, because that's really where a lot of that carbon is getting stored in the soil. So it's really in this first foot or so that we're really seeing a lot of that soil carbon that's newly being sequestered into soils.
Next question, was there any thought of using carbon dating to determine the age of the carbon deposit?
No, we had not thought of doing that. I'm sure that would be a good thing to look at for future research, because I know they're still looking at the CRP fields and still continuing this research.
Definitely. Yeah, that would be really neat. Does decreased bulk density result in increased water infiltration into the soil?
So yes, that would result in increased water infiltration. It's going to get the water down there deeper into the soil for those roots and allow them to grow deeper in there.
Awesome. How do you know Phalaris was not part of the CRP planting mixture on purpose or inadvertently as a seed contaminant?
So it could have been in there inadvertently for sure. So these certain conservation practices do have specific requirements for what seeds and kinds of plants they are supposed to plant. And as Phalaris is an invasive species, that would be something that they would definitely be trying to keep out. And they actually try to manage to keep those things out of the fields, but it's very difficult to do.
Thanks for that answer. Listener named Tyne says, how long is typical tenure in this program? Do landowners have a specific time commitment to retaining the fields in this carbon program, or is it a year-to-year decision?
So they generally do have contracts that last longer than a year. They have been going up to 15 years recently, but they do have the option to re-enroll these fields. It just doesn't happen very often, as a lot of times they go back into crop production.
Thank you. And an attendee named Murray is wondering if you have any references for different soil types as to their capacity for maximum carbon sequestration and what that maximum might be.
Sure. So if you look online at the USDA NRCS soil survey, you can just type that into Google, and when you go in there, you can find all the different information that they have for different soil types, which we'll talk about how much carbon they're expected to sequester, if it's more clay or loamy, and all those kind of questions.
Great, thanks for that answer. It sounds like a great website.
It is.
And an attendee named May is saying, what is expected soil carbon for a soil type? And maybe she missed it. Where does that red line baseline come from?
So that baseline was from the soil survey from the USDA. So they go out and have people that go out and look at all these different soils and do surveys, and they'll average all these soil types together and find what that average carbon is expected to be for that soil type.
Thank you. Thanks for clarifying that. And Ross says, there is an assumption that long-term perennial plant communities do not sequester and store as much as annual cropping systems. Is this valid logic?
I'm not sure I quite understand the question.
Do you think that like a long-term perennial plant community would store more than in like an annual crop?
Oh, sure. Definitely. So since that those plants would be staying there and not exposing that soil to the air and that wind and water erosion. They're going to be holding on to that soil and keeping those anoxic conditions more than a crop field would that's being plowed under year after year.
That makes sense. Thank you. And Tracy is wondering if you have any, or if you know of any CRP sites that would be similar to a restored roadside meadow, and if there are any estimates of soil carbon for those.
I do not know, but that is a good question.
Yeah. And then this could be the last one, and it's a bit of a doozy.
Okay.
Do you think your data could be used to create values for farmers to use in carbon markets and get some financial benefit from the carbon stored?
Sure. So actually, one of the new goals that the USDA will be looking at is carbon sequestration as a goal of the CRP programs. So they might be looking at actually making that a specific program to sequester carbon and then allow them to have those yearly payments in exchange for keeping those fields and keeping that carbon in the soil.
Awesome, that's really neat. Well, we're right at 1 o'clock and we just made it through all the questions.
Great.
I kind of gave them to you pretty quick and you handled them pretty quick. So thank you for that.
Oh, sure.
And we've had a few people type in and say it's very well presented and good research. And I just wanted to kind of reiterate some of their positive comments there. So thank you so much, Destiny, for taking the time to share your master's research with us today. It was really, really interesting and a great fit for our Prairie's Got the Goods week. So thank you.
Oh, sure. Thank you for having me.
And to all of our attendees out there, thank you so much for catching our last webinar for Prairie's Got the Goods week. If you're interested in more webinars, check out our Native Prairie Speaker Series. We have a monthly webinar going on throughout the year. And you can also check out our YouTube channel. We have some really awesome webinar recordings there as well. And when you leave today's webinar, there'll be a quick 1-minute survey, and we would love it if you could fill that out and share some feedback with us. We can keep our webinars going into the future. So with that, thank you so much, everyone. Happy Friday, and have a great rest of your day.